Hello guys, it's Doc Kyle once again, and tonight we're gonna talk about immunohistochemistry. So immunohistochemistry is simple, in the sense that it's just a mash-up of words. Immuno meaning we use the immune processes, our antigen and our antibody binding. Immunocyto meaning we apply it to our targeted cells, and chemistry. So the chemistry part is more of the chemical color indicator, no? Color indicator na part sa ato ang reaction. So what is the antigen antibody um, at kaning bond, no? So the antigen antibody bond is the most specific bond, type of chemical bond in chemical reaction, rather, in whole of biology. So it's quite unique in a sense that the antibody is only can only recognize this type of antigen, no? So the antigen in itself only means, the whole name of the antigen is antibody generating. So in a sense that if you are exposed to an antigen, and it's expected to produce an immune response leading to a production of your antibody. If it doesn't, then it is an inert compound. Thus, it will not create any immune response. So okay, what are the types of antigens? So the types of antigens, it can be carbohydrates, protein, or lipid. The more complex the antigen, the better it is to stimulate an immune response. So the thing that most immunologists, even in the past, no, even currently, try to discern is what first happened, no, where was the antibody first created, or was the antigen first, was the one to elicit the production of the antigen, because the antibody, rather, because um, you will talk about this more in immunology. But before an antibody can be produced, the antigen should be processed by your antigen processing cells, such as your histiocytes, your alveolar cells, and then they will be the processed antigen will be um, redirected to your lymph nodes, in which the B and T lymphocytes will get to work making an immune response against that antigen, no? So in the context of our discussion about immunohistochemistry, we are trying to utilize that feature of biology, that the most the most specific chemical reaction is the antigen antibody antibody kato binding, is when we see a cell, probably an atypical cell, no, a tumor cell, a neoplastic cell, and we don't know where it's from. We don't know if it's from the breast, no. It most of the time we do immunostaining for, mga metastatic, no. We also do for management purposes. So for example, we have breast cancer, no? And then in this breast cancer, there are like I said early, in the previous lectures, different types of tumor cells are sensitive to different types of your growth factors. For example, your breast cancer usually is kanang sensitive to your estrogen, because estrogen promotes the growth of the ducts and the glands of the human normal breast. So if there is a neoplasia, or a tumor that will arise from the breast, what will happen is they're very sensitive to this estrogen. And then, even though gamay lang tong estrogen na mudikit sa ila, they will proliferate rapidly, no? So that's what happens. They're sensitive for your estrogen receptor. And that's why when there's a breast specimen for immunohistochemistry, and then we see that the tumor, the breast mass, the cancer is sensitive to estrogen because it's stained positive for your estrogen receptor, um, then we can give drugs to inhibit the attachment of the estrogen, which in turn will eventually kill the tumor because there is no more signals for it to proliferate. So, always remember, like I said earlier, an antigen is anything that can induce an immune response, no? So, and an antibody is that is the reaction to that to that antigen. So, if I were to make an antigen against the if I were to get the estrogen receptor from tumor cells, no, estrogen receptor from tumor cells, and then I would introduce it to another animal or another cell that is which makes it foreign to the body of the animal. I will take the estrogen receptor from a cell, then I will introduce it to the spleen maybe of the rat, no? And then always remember that in immunology, if the body does not see you as the self, no, not part of the self, then automatically you are an enemy. There is no in between. So any foreign object, any foreign compound, the body will always mount an immune response against it. So ideally, because there are some cases na sa high dili niya mabatyagan. So anyway, going back, once I put that estrogen receptor that I isolated from the tumor cell, no, and then I put it in the spleen of rats, the spleen of the rat will make antibodies against it, no, and then I will harvest those antibodies from the rat serum, and then I will attach a kanang detector compound that in which like our horseradish peroxidase, which we will talk about later. What happens is when I take that antibody against the estrogen receptor that I harvested from the rat, then I will use it to attach to a piece of tumor, breast tumor, no? And then I will add a reactant. So once if that breast tumor has estrogen receptor, the antibodies that I harvested from the rat will attach to it. And what, di ba I attached a color indicator sa ato ang antibody, and then I will add another color reactant. Then in the slide we can see that nag-stain ang mga estrogen receptor, no? Kay tungod atong antibody na atong na-harvest from the rat, no? So therefore we can diagnose the patient's mass as estrogen receptor positive. So if we can give the patient kato anti-estrogenic drugs, then we might help in the inhibition of the growth or the proliferation or eventual death of the tumor, no? In turn helping the patient survive longer. Oh, kato, that's the gist of immunohistochemistry. There are mountains of information that I would like to impart to you regarding immunology, as it's one of my favorite subjects, but kato, short atong time and I'm trying to be concise, no? So immunohistochemistry, let's roll. So kato, no? So next slide. So as you can see, sa ako ang top, I don't know, top corner na ako ay GIF. That's your monoclonal, that's your antibody. Ito mo dikit siya back. That's the most specific, no? So remember, your antigen is your epitope, meaning it is the key, and then your paratope is your antibody. In the fragment of antigen binding, you will find your hypervariable loops. So kato, antigenic determinant site. So, next slide. So, immunohistochemistry, tanawan ninyo ng GIF diha. This is the primary antibody. Usually, the primary antibodies are kanang kato lagi harvested from the rats before. But right now, we're using immortal cells, no? Immortal cells. And then, what happens is we create a hybridoma. But I digress. Let's talk about that later, no? So, immunohistochemistry is the technique to visualize recognition of antigen present in the tissue with the help of the corresponding antibody. The basic principle of immunohistochemistry is to demonstrate the specific antigen in the cell by applying the corresponding antibody to the antigen-antibody reaction. The antigen contains the epitope or the antigenic determinant site that evokes specific immunologic response to develop an antibody. The antigen epitope site and the antigen binding site have complementary geometrical and chemical features. This is responsible for the antigen antibody reaction. So what happens? Why are there daghan pagpakapin in our GIF na sa taas apart from our primary antibody? Our primary antibody is what I discussed earlier, the one that has been exposed to the tissue. This primary antibody is commercially produced. No? It's produced by big laboratories and they use lots of techniques which we will talk about later, like your hybridoma. No? So anyway, so what happens is when the primary antibody attaches to our target antigen sa ato ang specific tissue na gusto nato mabalan kung sa yung pulos, the FC portion usually is exposed. So we will add another reagent, or secondary antibody, which in turn has the katong akong bio same like ani na siya avidin ata ni. Yeah. So streptavidin, which will attach to the FC portion of the primary antibody. And then katong secondary antibody ang mag facilitate sa mag-color na siya. So what is the purpose of this? Ngano daghan man siya? Ngano kanang dili man siya pwede idikit na lang mismo sa primary antibody? It's because the primary antibody, number one, is tailor-made for that tissue or for that antigen, no? Kung buhaton nimo na siya tanan unya, medyo it's quite hard to attach the color detector component sa in an antibody. Tapos buhaton pa jud nimo, tailor-made pa jud tong FAB site sa imong primary antibody, then it's quite tedious and it's not cost effective. So what you'll make is a primary antibody that is specific for that type of tissue, and then you'll just make a generic secondary antibody that attaches to the FC portion of any antibody, non-specific binding, no. So apart from that, it makes the reaction more sensitive in a sense na because duha ang fragment of antigen binding sa imuhang secondary antibody, pwede duha ka primary antibody iyang i-bind to, no. So meaning mas dako imuhang makatch na mga antibodies, no. So mas daghan makatch na antibodies tong imong secondary antibody. So meaning unlike if isa lang siya na modikit sa isa ka antibody ay na antigen imong primary, dili siya ato ka sensitive. But if you're gonna use a secondary antibody, the net is much wider. So kato. Next is kato. So antigen is a substance producing of an immunologic response. Is called antigen, no? Kasi mao gani sa name gani niya, antibody, antibody generating mo na siya, no. So the antibody is also known as your immunoglobulin. It is produced by your plasma cells, no, not by your B cells. No, your B cells are the precursor for your plasma cells. Your plasma cells are your activated B cells. So the first antibody that will be produced by your plasma cells is your IgM. So if you have an acute infection or an acute immune response, the type of antibody that will be produced by your plasma cells is your IgM. So if you have an acute COVID infection, most probably the antibody you will produce is still IgM. So what are the drawbacks with IgM? Always remember IgM is a pentamer, and then what binds those five antibody molecules together is your J chain. No? So this pentamer is very good in activating your complement, and it's very good in coagulating, clumping together cellular na mga components. So because of course five man siya, so mas daghan siya mapaklamp niya dayon iya ang mga RBCs ay ang mga cellular na mga offender, mga cellular na mga foreign na mga cells, no? So kato. So example, if you have IgM in your na antibody sa imuhang blood, and then naakay ka nang na-transfusan ka og wrong na blood type, no? So the IgM will clump together. So mas daghang maklamp together imuhang IgM na antibody. In contrast to your IgG, which signifies that the switch from an acute infection, well, not necessarily acute or recent, to a more kanang nag-start na siya og resolve or maybe na prolong, because IgG usually signifies long term. They say immunity man good, but I prefer long term protection against the type of antigen or offending compound or pathogen, no, na exposed. So if you already have IgG sa imuhang example COVID na test, no, then most probably you have been exposed before or you have been vaccinated. So remember, IgM is the more acute or current infection, previous recent. So the catchwords are recent, current, no, for your IgM, and then your IgG is the previous or vaccination, previous or vaccination. Vaccination is by all means is just a type of kanang infection kanang infection without infecting, no. So they just provoke in all sense, sa tanang mga butang, no, vaccination is just a provocation of your immune system against a certain type of pathogen or bacteria or virus, no, same with your COVID. So this term from the switch from IgM to your IgG is called class switching. So nag-class switch na siya from IgM to IgG. There are multiple there are diseases that inhibit this class switching because there are certain enzymes needed for it. So kato, I digress. So each immunoglobulin is composed of a pair of light chains and a pair of heavy chains. So and then each light chains are two types, your kappa and your lambda, and then your heavy chain is alpha, gamma, delta, epsilon, and mu. So the type of class of antibody is dependent on the heavy chain. So remember, it doesn't matter what your light chain is, your heavy chain dictates the class of your antibody. The two tips of Y are also known as your antigen binding site, and each arm of Y is The base of each arm of Y is the hinge, and it's the flexible region of your immunoglobulin, no? So hinge in a sense na pwede siya so it can move, no? It's quite fluid in that area because of the high amount of proline. Correct me if I'm wrong. Proline, amino acid in that. Always remember your immunoglobulin is a protein. It's one of the two proteins that is not produced by your liver. All proteins, either your globulins or your pleated proteins, are produced by your serum proteins rather. All serum proteins are plasma proteins are produced by your liver, except your immunoglobulins, which are produced by your plasma cells, and your um, von Willebrand factor, which is produced by your endothelial cells, no? So your remember that an amino acid, a protein, has two ends. Your amino group, no, that's where your nitrogen is, and your carboxylic end, so COOH. Your amino group, that is where your fragment of antigen binding is, and your carboxylic group, no, is where your fragment of crystallization is. So the fragment of crystallization, they ngano naman yung crystallization, no? Why have we carried this? Because in the previous, when they studied your immunoglobulins, we used crystallography. We make them crystals. We add compounds to make them crystals, and then we can study them if they're a crystal na. Because remember, your immunoglobulins are soluble ng mga substances, meaning matunaw. Not necessarily matunaw, but they're freely soluble sa imuhang blood, no? So in order to study them, we need them to be solid. That's why we try to do crystallography. So kato, no? So your IgG is a very kanang it's the ideal antibody because number one, it has a lot of functions. It can neutralize, meaning once nidikit na if I have a pathogen ko, let's just say this is COVID, corona, and then in this my fingers are the receptors, no, of coronavirus. Remember, viruses attach through receptors sa imong cells, and when they attach, they gain entrance within the cell, no, thus infecting them. Always remember, your viruses are obligate intracellular organisms. We don't actually know if viruses are alive or they're just a piece of genetic information, no? So anyway, what happens? The IgG will block the receptors of the coronaviruses, inhibiting the entrance into the cells. Kung dili sila masulod sa imong cells, dili na sila ma-infect, no? Kung wala kay infection, dili ka magkasakit, no? Alright, goods. So, next slide. So this is how you produce your commercial na antibodies, because no, you can't manufacture it because like your antibodies cannot be kanang produced tungod kay dili man guni siya mabuhat lang sa laboratory, no? It's quite a complex. The mere fact, no, we need to be grateful to the Lord, no, gibuhatan ta niyang antibodies. The mere fact na mudikit ang antigen sa antibody is such a very it's hard to fathom no? Because minusya kaya na siya ng molecules, no? So dili ni siya pareha sa dagom na mosulod lang ang hilo, no? So uso ni siya kanang una mo siya susi nya lock nya kakita ka ng abog, mas gambay ba pa sa abog. it's very complex, and then it's dependent on multiple variables, no? So this hybridoma monoclonal technique, kung mo na akong giingon sa inyo ganiha. So we get antigen. We usually get antigen from your control na mga tissue. Example, like I was talking about ganiha, estrogen receptor, no? The estrogen receptor is also positive in your normal endometrium. It's the same receptor as the normal estrogen receptor. is it's the same receptor you can see in your kanang breast cancer na estrogen receptor positive, no? It's the same receptor but different ilahang location na ay pathogenic sa imong breast mass na apoy kanang physiologic na sa imong endometrium lang. So we get this antigen, like I said, we tusok it in the ilaga, no, na si Mickey Mouse, no, and then mag-produce na si Mickey Mouse o kanang antibodies against that receptor kay foreign lagi siya. And then we isolate, actually hardcore nilang ginabuhat din, no, kuhaon nila tong mga plasma cells na naga-produce og actually hardcore kayo na procedure na kuhaon nila tong mga plasma cells sa rats. So masyak mo na pwede dahil na mahitabo, pwede na siya mahitabo, and kani siya, it's really quite sad, no, because mo jud kailangan jud mamatay sa ilaga, no, kay alang I don't know kung kaya nila ma-spare ang ilaga, but kato kuha nila ang mga plasma cells usually naman sa spleen sa ilaga, so they have to kill the rat, open the spleen and gather the plasma cells, no, viable na plasma cells, buhi pa ni siya na plasma cells, no. Kanina plasma cells nag-produce ni siya og antibody against the antigen that was injected. In our example, the estrogen receptor, no. So anyway, kato, and then we usually fuse it with a tumor cell. So what is this tumor cell? Always remember your tumor cells are freaking immortal, no. Di na sila mga matay hantod mahutdan sila pagkaon. So kaning mga scientist na bright kaayo, so diha ka makaingon pa unsa ka nila ni hunaan sa una. So nadyoy mga parts sa science right now na it's really hard to walk away from, no, nagahimo jud tag human experimentation sa una. Even though ang mga German sa una tong World War II kanang hardcore kayo lang gibuhat, no, wa jud lami ba, di jud makaon, but we still can't deny the fact na na-advance jud nila ang medical practice. I don't know kung nani relation, pero kung huna-una mo kinsa goy tao na maghuna-huna na magkuha og tumor cell, no, unya i-combine niya sa plasma cell sa ilaga para maka-produce og daghang antibody, no, lisuda po na huna-huna, no, but still it's the process that we're doing right now. So if you fuse those plasma cells na na-harvest nato sa ilaga into a tumor cell, that is now what you call your hybridoma, and your hybridoma is now immortal and will produce antibodies forever until eternity as long as naa siya'y nutrition, no, kumpleto yang nutrition. So these antibodies is what we use for immunohistochemistry and or, no, kahinom pumapaakan mo og miming or any dog, depende sa classification sa inyong animal bite, kung unsa inyong hatag, na example nagdugo-dugo siya kay lang. There are two types of vaccination, no. We have our passive vaccination and our active vaccination. Active and passive are in relation to our immune system. So if we are given an active vaccination, that means we are provoking our immune system to produce our antibodies. What are the benefits? Benefits, it's quite cheap because we just give the antigen. The antigen is usually a dead or kanang immobilized or lagi dead or parts of kanang bacteria or virus. Same with our COVID, our tetanus, no, our pneumococcal. Those are only kanang attenuated na mga vaccine or ay na mga microorganisms. Attenuated meaning weakened na kaayo. Not enough to cause sickness, but enough to create immune response. Then there is what we call our passive immunization. Our passive immunization can either be physiologic or kanang it can be natural or kanang man-made, no, or artificial. So our passive na immunization na kanang physiologic or kanang natural is the passive immunization that we get from our mudracels, no, when we were still a baby. Inside the placenta are the preformed IgG of our mother will pass through the placenta and go into our bloodstream as newborns. So, as fetus pa, no? And then, those antibodies will circulate in our blood. So, katong antibodies, dili kita naghago ato, atong mama naghago ato. So, another point of contemplation na pasalamat dito sa atong mga mama kay even though fetus pa tagin ampingan na dito nila not just the kanang sa ato ang physiologic nutrition needs sa ato ang unsa pod immunity no ginatampingan dito nila. Anyway, going back, so passive siya in a sense na walay provocation of the immune system or activation if the immune system is done. We only dawat limpyo this preformed antibodies, no? So it's passive. In artificial or kanang man-made, what will happen, no, if what are the benefits of active immunization? It's cheap, and then usually cheap niya. Problema mong good aning active immunization, mao jud dapat ning ideal, and then we should get it before we get the sakit ideally, so that when we are exposed to the pathogen, ready na the antibodies to fight. But it takes time to produce these antibodies, on our own. So that's one of the drawbacks. We need time to produce these antibodies. But in passive, since giyatagan ng antibodies diretso, they are ready to fight dayon. So kung lala kayong paak ka ng sure ball na nadyo'y rabies tong nipaak sa imo na shih tzu, no, nagtulo-tulo jud ang laway sa baba, no, gipaak jud imong aping, you really need to have pre-made or kanang ready to fight dayon na antibodies, and that's your pre-made antibodies sa imong unsa na passive immunity. So itusukan na dayon kag antibodies na ready to fight para i-fight na dayon niya tong rabies na virus na sa imong aping, no? So that's your passive immunity. Drawback of your passive immunity, no, it's very expensive. Kay, no, gina-harvest nila ni dili sa ilaga, sa mga kabayo. Usually ako makita kabayo, no, sa kabayo nila ni gina-harvest. Of course, there are other more expensive means to harvest antibodies, but makakita mo na kung mag-medicine man mo, makaduty mo ng mga animal bite center, kita mo na gikan mga equine, no, meaning kabayo gikan ng mga antibodies na siya against sa rabies, but giharvest na siya gikan sa kabayo, no. So anyway, so that's your active and passive immunity. So that's so to sum it up, that's your hybridoma and your monoclonal technique. And then your polyclonal antibody, polyclonal is generated from different B lymphocytes to form different epitopes. So, and then they are generated from different clone of B cells, and these antibodies known are polyclonal. So, simply put, your hybridoma, isa lang ka cell iyang origin, gidikit lang siya sa imuhang tumor cell mo na to. Monoclonal, isa lang. While your polyclonal, of course, daghag B lymphocytes ang gigamit, no? So, affinity versus avidity. Affinity refers to the strength of a single antibody-antigen interaction. So, affinity is high, no, in your IgG. So, the avidity refers to the total interactions combined. Excuse me. So, why is the affinity of your IgM is lesser than your IgG? Because dili tanang site sa imuhang IgM magdikit sa antigen, no. Kaya of course dagan kay siyang sites, but dili tanan mudikit tanan, no. Makagets ka? Pero overall mas dagan gahapong sites ang mudikit versus sa imuhang IgG, which duha lang iyang sites. So most probably dakong chance na since duha lang man siya mudikit jud siya tanan. That's why it's a high affinity, meaning all of the binding sites will be used up. Versus your avidity, it doesn't matter if all the sites are used up. It only matters how many sites are used up, no? So it's 100% versus 80%, but your 100% is only 2 versus your 80, let's say 80% of your IgM, pila ba na? 80. My gosh. 8, oh. Okay, 5 times 2. Oh, so 8 is still greater than your 2. So that's your avidity, the total, no? And then your affinity is your percentage. So valency, the more valence the antibody, the greater is the avidity. Affinity, kato. So antigen specificity. So ako makita anin na gwapo na i-PowerPoint na slide. So so the antibody specificity indicates the precise detection. Like I said earlier, it's the most precise, the most specific chemical reaction in all of biology. The antibody antigen reaction. So kato. So detection system. So the direct method, the primary antibody is tagged directly by an enzyme or fluorescence. The antibody should be specific for the particular antigen, otherwise the specific staining, non-specific staining may occur. So tanawon nimo ng picture, no? Oh, so here we have our sorry. So we have our antigen. Then we have our antibody. So we already have fluorescein here, our indicator. So mawala siya, isa lang siya, direct. So detection system, it is impossible, no, to detect an antigen antibody reaction by light microscope. So therefore we really need a visualization system. So in the direct method, the primary antibody is directly tagged or nana ni nakadikit daan detecting method, which is your fluorescein or your enzyme kaning mark sun sun din hino. So anyway, ito. So the antibody should be specific for the particular antigen, or otherwise non-specific staining may occur. It's a rapid because isa lang nimo siya ka pag dikit sa antigen antibody mo hayag dayon siya. So different primary antibody should be labeled different for antigen and low sensitivity. So like I said, dili siya cost effective kay kada isa ka antibody na imong buhaton, you will painstakingly dikit the fluorescence, no? Kani siya imuhang kana imuhang fluorescent. No, so una-una kada himo nimo kada harvest nimo na sa ilaga, no, dito sa hybridoma, butangan pa nimo gana na pod, sula no. So kato, so the next slide. So our indirect method, no, so versus our direct method, sa atong indirect method, if the meaning aning indirect method, dili ang imuhang primary antibody ang directly mag-stain. We use another antibody or secondary antibody na kato siya ang mag-stain or mag-colorize. So, we usually use our peroxidase, anti-peroxidase method, no? So, kato. So, like I explained earlier, bali na ko niya akong nawong, no, kay para makita ning siya. So, this is your tissue antigen. This is your primary antibody. In the direct, pag dikit ana niya, mag-color dayon na siya, na siya color. But in the indirect method, there is another another antibody that will detect that will attach to your FC portion sa imuhang primary antibody. And then mo na to ato ang streptavidin biotin or our enzyme complex or our kaning horseradish peroxidase, and then we will add a chromogen na mag-color. So what are the advantages? Of course, like I said earlier, it's more sensitive in a sense na kani mong secondary antibody, pwede pa ni siya modikit og another primary antibody na nag-attach sa imuhang antigen, leading to mas daghan iyahang nadakpan na color na unsa na antibody na nindikit. Because kung primary lang siya, isa lang siya diha, weak lang kaayo iyahang staining kay isa tagsa lang man na siya. Always remember, even though mag-stain ni siya, as long as dili matapok together ang iyahang mga antibodies na nagdikit, dili kay siya discernible. It's quite faint. So if natapok sa secondary antibody nimo ang mga primary na antibody, and then mag-stain siya, meaning mas dark siya, mas klaro siya, no? So high degree of sensitivity, the peroxidase anti-peroxidase is 1,000 times more sensitive than the indirect conjugated method. So kato. So sige, a single mali dahil na advantage ay rather dini dahil sa indirect method na advantage, no? Dini dahil na advantage dapat. So sorry wala to ninyo nakitan, go back ko. So primary antibody in your direct method, dikit na siya diha, naghayag na siya. But if your secondary kani siya, it can dikit pa to another primary. So duha na iyang madikitan na primary antibody. And then mudikit kani siya imuhang kanang nakadikit din sa imong FC portion ang imong streptavidin or enzyme complex na siya ang mo-color, no? So like I said earlier, you really need to have mas gwapo kung mas daghag ma-group together imong mga primary antibody, no, na mabind sa imong secondary para mas maklaro siya. So a single conjugated antibody can be used against different primary antibodies. Isa ni siya atong advantage na dili na ka magdikit og kanang marker, kanang coloring ay kanang identifier sa imuhang primary antibody kay mahal siya, nya gasto, no? So kato, higher dilution of primary antibody can be used, large amount of secondary antibody can be produced against primary antibody. That is because ang target lang sa imong secondary antibody is the FC portion of your primary antibody. Dili na siya kailangan tusok na pud sa ilaga, no? So kato, discard. Next slide. So just read this, guys. Dili man po ko strict to. Naara diha. So these are the types of your secondary antibodies, no? The techniques in your indirect staining for your immunohistochemistry. You have your peroxidase antiperoxidase method, then your avidin and biotin, and then avidin biodin conjugated alkaline phosphatase. So kani sila manggod there every year na jay bag-o na pud mo gawas manggod na na gina-market sa ilang mga produce. So I'm not that keen, no, to, of course, tried and true na ni sila, tried and tested na ni sila ng mga test, but kato kanang kada tuig manggod na imong gibag-ong style na pud lagi daw na mas better lagi daw, but I digress. Kani sila ang mga tried and true na mga tests, no. So please read on it lang. So control, importante kayo ang control nato. So what is control? So control is the, okay, so we go back to lab management. Control is your to check whether you have done the test correctly or incorrectly. Standard is the chemical or compound or material that we use to calibrate the machine to set the boundaries between high and low. That's our standard. And our control, we usually have a negative or positive control in immunohistochemistry. Mag-ingon ta kung positive control siya, meaning nag-stain ba jud ato ang slide, no? So sakto ba ang pag-stain? So think of it like this. Ang control kay kana siya ang wait, magkita lang ko picture. Alright, game, no. So mao ni siya usually ang control, guys, no. So this is a prepared slide, no. This is the end point when we're gonna process our immunohistochemistry. So this is a slide, and this is the specimen that we katong gikan jud sa patient, gikan sa iyang breast mass, gikan sa iyang ilong, and this is the control. So what will happen if we're gonna look at the slide of the patient in the microscope, and then we're gonna compare it to the control. The control is usually positive. So meaning, if makakita tag staining na same intensity with your control, meaning imuhang giran na procedure, imuhang staining, it's valid, no? Guwapo ang iyang pagkaran. But if pagtanaw nato, if nahatag sa to na slide, ang staining sa imuhang control kay kuspaw kayo or wala jud siya nag-stain, then wala murag ma-question nato ang viability po sa staining sa ato ang specimen. That's the purpose of your control in your histochemistry, no? To check if the procedure has been done adequately and walay mga lapses, no? So there are a lot of kanang mga problema like nahupas na ato ang reagents, no? Or improper, nagka-power outage and such. So this is the procedure of your immunohistochemistry. Ana siya. And these are the types of your immunohistochemistry. So thank you guys once again. Thank you guys once again for gaulan manggod. So basig dili gwapong audio for listening to me. This is my last lecture in histopath. We still have kanang face to face or online Zoom class, and I hope to answer more questions no or kanang mga to explain it further. Immunohistochemistry is very this is the future, not necessarily the future. Genetics is the future of histopathology, and immunohistochemistry is more of the present. But currently in the Philippines, medyo kanang delayed pa kayo ta. So hopefully na magpatho ninyo, and then hopefully makabalo mo more about histochemistry. So thank you for everything guys. So ay mo kaulaw na mag PM lang nako, and good night, God bless, happy studying.